Hello everybody, in this video I'm going to talk you through some of the important aspects of digestion and how we can apply them to an exam question. The first two questions are fact recall questions, and this means you just have to learn this material. In the digestive system, it's the stomach that produces hydrochloric acid, and it does this so that it is a defence against disease that might get down in your food into your stomach. To deal with the acid, the lining of the stomach is specially adapted so that the acid doesn't agitate the stomach wall. On the diagram, this is the stomach. So part C is the stomach, and that's what we should tick. For the next question, we have to say which organ produces bile. Bile is made in the liver and then stored in the gallbladder. B is the liver. Bile is designed to help emulsify fats, so it binds to the fats and breaks it up into smaller pieces, meaning that enzymes can work on it much more effectively and much more quickly. So B is the correct answer. This question is also a fact recall question, so just something that we have to learn. So digested foods are the broken down products get absorbed into the blood in the small intestine, and the small intestine is specially designed to do this. The small intestine is at E, where I've shaded it blue. Inside the small intestine there is a specially adapted lining, and you can see what that lining looks like in the diagram that I've put just next door. So we call it villus or villi for plural, and these are like extensions and folds within the lining of the small intestine. Having it all folded gives a bigger surface area, so we can maximise how much can be absorbed into the blood. You'll also notice in the cross section that there's lots of capillaries going up into those folds, and the high network of blood vessels that are surrounding them maximise the amount of digested products that we can get directly into the blood and gives a short diffusion distance. However, we can only absorb very small things across the wall of the villi, and so they have to be broken down. Things that we eat, starch, protein, fats, are big biological molecules, and they wouldn't fit across that wall. So we have to make sure that we break them down first of all, and that requires enzymes. This leads us nicely to talk about the activity of enzymes. Enzyme activity really refers to how fast does the enzyme help to break down the thing that it's trying to break down. So it could be the starch, for example. Remember that enzymes are specific, so a protease enzyme will only help to break down proteins. Amylase will only help to break down starch, and lipase will only help to break down fats. So in this particular question, it's talking about protease, so we know that that's going to be acting on proteins. Just as a reminder about enzymes, here's a diagram, and you can see there's a couple of important things to note about the enzyme. First of all, it's made of protein itself. The second thing to note is that it has a very specific shape called an active site, and this is where the substrate, that would be, for example, our protein or our starch, is going to fit into in order to be broken down by our enzyme. Now, enzymes are really fickle. They like to have very particular conditions in order to work. Some enzymes like to work at hotter temperatures than others. Some enzymes like to work at particular pHs, and each enzyme has its own set of very particular rules that it likes to follow. Put an enzyme into the wrong conditions and you can damage the enzyme by changing the shape of the enzyme, which would alter the shape of the active site, and that would stop the substrate from being able to fit. The alternative is that you can just slow the enzyme down, so it just doesn't really work very efficiently, it's not moving around very quickly, and it's unlikely to actually bind with a substrate at all. In this question, they're talking about pH, and they've got two different enzymes, X and Y, and they're looking at how fast the enzymes work in different pHs. For this question, you need to know what the word optimum means. Optimum means the condition under which the enzyme works at its maximum rate. So for enzyme X, we can see that that's here. And for enzyme Y, we can see that that's here. So if I want to work out the optimum pH for enzyme Y, I need to read down from the optimum. My line is slightly to the right there, but it should be 7.5. As we move away from the optimum, either lower or higher, so down to 5 for enzyme Y and up to nearly 10, we see that the activity of the enzyme drops off quite quickly. You'll also notice that enzyme X and Y are totally different. Enzyme X would much prefer to be at a much lower, more acidic pH, pH 2. So this goes to prove that enzymes have their own very specific set of conditions that they like to work within. This is another fact recall question, and we just have to learn these. So you need to know where each of the enzymes can be found. This question is asking where in the digestive system might the two protease enzymes be produced? Well, we've just seen that one of them has a very low pH, pH 2. pH 2 is very acidic, and we know that the stomach has stomach acid, so it would make sense that we would find it in the stomach. For enzyme X, there's only one option where enzyme X is in the stomach, and that's this bottom one here. So let's just check that enzyme Y makes sense. Enzyme Y preferred more of a 7.5 pH, so that means that that would definitely be found in the small intestine, where it's much more neutral, is much less acidity there, and if anything, it's slightly more alkaline. So that would make sense. So the stomach for X and the small intestine for Y. This question required you to link together a few things. You're understanding that enzymes are found in different places in the digestive system, and that those different places have different pHs, and then using the graph information to link which enzyme would go where. This question is testing your knowledge of the active site. The active site, as we've seen already, has a very specific shape, enabling it to latch onto or bind with one particular substrate, and this makes enzymes specific for different substrates. Now, even if you didn't know that, you could get the marks here just by viewing it as a shape sorting activity. So you need to imagine figure 5 turning around and then fitting onto each of these shapes, and you need to identify which shape that would fit onto. Here's the shape turned around, and although it would fit on these two diamond shapes at the end here, there's nowhere for the rectangular shape, so that's not a complementary fit. Let's try the next one. Okay, in the middle here you can see, particularly if I make this slightly larger, that potentially we have a match. So the diamond shapes would fit into those holes there, and the rectangular shape would fit here. So B would be our answer. This is another fact recall question. So you need to know the name of the theory for enzymes and substrates coming together and binding. It's known as the lock and key theory. As you can see from the diagram, a key has to fit into a lock, and it's always a specific shape. You can't use any key in any lock. They have to fit. The shapes have to be complementary. Now, it's important to note that the shapes are not the same. The lock actually has a hole or a negative shape, and the key actually has a shape that's going to fit into that. So they're not the same shape, but we use the word complementary. It's the same with the substrate and the active site. You can imagine that the active site or the enzyme is acting like the lock and the substrate is acting like the key. So this answer should be lock and key. For this last question, we need to actually start explaining. So this requires some understanding, not just learning. So in the diagram above, in the top part of the diagram, you can see that the substrate fits exactly with the active site. If I change the pH though, that's going to have an impact on the bonds that hold together the enzyme. And it's going to change that so that it changes the shape of the enzyme. And this will lead to a non-complementary shape. So now the active site does not fit the substrate, and that means that it can't fit together, and so it won't work any longer. For this answer, we can simply say that changing the pH can change the shape of the enzyme, affecting the active site shape. And this means the substrate can't fit into it anymore, so the rate of the reaction decreases or may even stop. This is the end of our video on digestion and answering exam questions about digestion. If there are some concepts in here that you've listened to and you don't understand, please approach your teachers as soon as possible so that we can help you. Thanks very much. Have a great day.